Hello again. This is Roger Koch Barr for the Bioelectricity course, and we're starting week 5 of our course. Thank you for being here. We have an exciting but different topic today. We're going to talk this week about currents in the structure. By analogy, we are talking about the track on which the train runs. Please keep in mind as you think about this topic that this is one of the 3 topics, 3, 4, 5, one of the 3 topics that could be taken in any order. So topic number 5 is independent of topics 3 and topic 4. So talking about the track is really fundamentally different as compared to what we have done up until this time. We are now, for the first time, not staying in place in one little patch on the membrane, but rather examining what happens as currents flow from one place to another. There are 2 aspects of this for which we need quantitative results. The first which I've called axial currents inside and outside, are the currents that flow from one place to another, either in the space inside the cells or in the space outside the cells. The second is a consequence of the first. At some point along the fiber, the fact that currents stop flowing inside forces them outside. At other points along the fiber or in a group of cells, the fact that currents start flowing inside means that current has to come through the membrane from the outside. So we want to know, what about these membrane currents that are forced to be present by the need to supply or to get rid of the axial currents? These are our 2 major topics for the week. So we have 2 groups of topics that I've divided as usual. In the first will emphasize that this week is a little different from what we've done so far. And in particular, we will have to get straight a bunch of little things. All these are just little things that we have to get straight in order to deal with the track rather than with the train. Or in the bioelectricity sense, they're little things we have to get straight in order to deal with currents moving from one place to another place. You won't find any of these very hard. It's not a matter of difficulty. It's a matter of having them in mind. Then in the second part of the week, this part, we make use of all those little facts, bring them together, find the axial currents, and find the transmembrane currents, which is really what our objective is. As we start this week, let's look at the terms in the equation that we have used over and over. We have frequently begun with the equation Im equals IC plus I ion. The total membrane current is the capacitive current plus the sum of the ionic currents for active tissue. In earlier weeks, we have, for a patch, examined the capacitive current. So that part is done. Last week, in an intensive set of individual segments, we examined the Hodgkin-Huxley model for ionic currents. So that part is done. This week, we take the third item in that trio, and we're saying the total membrane current. This comes from the neighbors. Let's examine this week and see how that's done so that our action potentials can move from one place to another. It's great news. Now, all of a sudden, our patch is not isolated from nearby patches. And of course, that's absolutely essential because we want action potentials to travel along a nerve so as to carry information from one place to another. We want electrical signals to move through the heart so that the whole heart beats. We want electrical signals to move from place to place within the brain as new thoughts are formed. So the functional value of electrophysiology is intimately tied to this idea of a patch being tied to its friends nearby. It's not isolated. Mathematically speaking, up until now, we've always assigned Im is equal to 0. Now, we did that because we didn't know any current that was coming from the neighbors. We just wanted to test what was happening in our patch in isolation. And the way to do that was to say, well, whatever the current is in the patch must stay in the patch, or in other words, Im is equal to 0. There's a little bit of exception there where we have introduced a stimulus current for a short while, but this really had nothing to do with the neighbors. You will find that when there are lots of patches, and there always are lots of patches in naturally occurring tissue, one patch has a strong effect on what happens in the next patch, and that patch on the ones beside it. So there is a chain reaction type of operation. Each one has an effect on the others. And mathematically, this effect of each patch on the next is expressed by changes in the value of the membrane current, Im. There's another thing. Up until now, we've talked about patches in isolation with very little need to talk about the specific structure from which they came. We've mentioned along the way frog muscle, squid axon, but by and large, we've talked about patches all alone in the world. Well, it's just not like that anymore. Things are different now. We're examining how one patch interacts with its neighbors. And to do that, we have to do that for a particular tissue structure. So I'll end this segment with this picture of some Duke campus flowers. As you know, flower bushes, one flower has an effect on the others. Bees travel from place to place. The whole entire process in nature depends not just on individual flowers, as important as they are, but on their interaction and their interaction with the surroundings so that there can be flowers this year, next year, and the year after. We'll see just how this works in the subsequent segments.